going elevating blood glucose level. كان ناقص الجلوكاجون ننقص البلاد جلوكوز ليفل نزيد الانسولين ننقص البلاد جلوكوز ليفل. اوكي صار هذه الميديكال ابلكيشن ده. So number one, by biochemistry understanding the mechanism by which insulin is released from pancreas. عليها الضوء هدايا الفارماسيست discovering new drugs by understanding the mechanism of release, mechanism of absorption, and mechanism. Okay. So insulin released from the pancreas. So what is the function, mechanism of insulin? Mechanism of insulin. Insulin is a water-soluble hormone. So cannot cross the cell membrane, yes? Because cell membrane is a hydrophobic phospholipids. Yes. So okay. This is cell membrane. There is integral protein. The integral protein for a receptor. It is tyrosine kinase receptor. اسم الريسبتور هذا tyrosine kinase receptor. Okay. When there is no insulin, the receptor consisting from two monomers, dissociated monomers. This monomer 1, monomer 2. No insulin. Okay. In cytoplasmic part, there is tyrosine. Lots of tyrosines. Also, there is kinase receptor, which can phosphorylate tyrosine. Yes? هذا هو التايروسين صح؟ Tyrosine, yes? Phosphorylation at hydroxyl group. Okay. So when the insulin binding at the extracellular part of receptor, tyrosine kinase, the tyrosine kinase monomers will be dimerized. So, dimerized. Monomer, dimer, binding. So, monomer 1, monomer 2, interacted. So, هذا هو insulin. Okay. When the monomers dimerize, the kinase will be activated. So tyrosine kinase becomes active. The tyrosine kinase will phosphorylate the tyrosine. Phosphorylate tyrosine. So binding, dimerization will lead to activation of the tyrosine kinase phosphorylating the tyrosine in cytoplasmic part of the receptor. When the cytoplasmic part of the receptor phosphorylated will active the protein insulin substrate. Insulin receptor substrate. Insulin substrate. It's a protein in cytoplasm. When the insulin receptor substrate binding with the phosphorylated part of the tyrosine kinase receptor becomes active or binding إلى بيخلي ال ARS active biological active compound. The active insulin receptor substrate will activate the phosphatidyl anisitol diphosphate kinase. عارفين ان انيسيتول phosphatidyl anisitol phospholipid located in the inner side of cell membrane. So جهة السيتوبلازم. Okay. Phosphatidyl anisitol diphosphate. Phosphatidyl anisitol diphosphate will be phosphorylated to phosphatidyl anisitol triphosphate. Kinase, addition of a new phosphate group. Diphosphate becomes triphosphate. Phosphatidyl anisitol triphosphate will activate protein kinase B, class B, enzyme, protein kinase B. كنا عارفين ان الكاينيز الفانكشن بتاعه phosphorylate. When phosphorylate AS160. AS160 protein, yes, protein inhibited. This protein will be inhibited. Okay, we have, you know, G proteins, G protein receptors. There is integral G proteins in cell membrane, and there is soluble G proteins in cytoplasm. Two classes of the G proteins: trimer G proteins and monomer G proteins. So monomer G protein is located in the cytoplasm, soluble. Okay, so G protein is a protein activated by GTP, inhibited by GDP. ولهذا سميناها G proteins. Yes, G proteins. So أقول G اختصار كلمة شنو؟ guanosine. Yes, so activated by guanosine triphosphate, inhibited by dephosphorylation of guanosine triphosphate to guanosine diphosphate. Yes, so guanosine. So أنا عندي gap G T B active. Yes, gap GTB gap. So it's a class GAD. It's a class of a G protein, a soluble monomer in cytoplasm. So GAD, okay. So hydrolyzes from GTB to GDB by GTBX. Yes. So active inhibition. وهذا kinase. Yes. So GAB GTB activating the glucose transporter 4. Glucose transporter 4, it is not located in cell membrane, but by the mechanism of insulin will be released from vesicle حوصلة. أنا موجود في حوصلة glucose transporter 4 by activation by GAB GTP will be translocated from cytoplasm to the cell membrane. Glucose transporter 4. So كل الجلوكوز transporters located in the cell membrane accept glucose transporter 4, which is located in the cytoplasm, and the insulin, very important for translocation or relocation of the glucose transporter 4 from physical in the cytoplasm to the cell membrane. So هذا هو الميكانيزم of action of insulin. For insulin activating the glucose transporter 4, which is located in cells of skeletal muscle and adipose tissue. So هذا هو decreasing blood glucose level. How? How? By activating the absorption or transportation of glucose from bloodstream to the adipose tissue and skeletal muscle. Okay. That's the mechanism of action of the insulin. Now the glucose located in the cytoplasm. هذا كله glucose from bloodstream to the cytoplasm. Okay. So the glycolysis started before the insulin release, but where? In all tissues except the skeletal muscle and adipose tissue. Okay? صار التيشو للترانسبورتيشن لا يحتاج انسولين, the glycolysis started before the insulin released from the pancreas. Okay? Okay. صار لما يكون في مشكلة في السنسيتيفيتي أو الانتراكشن between insulin and tyrosine kinase receptor, it is diabetes mellitus class 2. Yes. diabetes mellitus class 2. لو يكون في defect في ARS diabetes mellitus. Defect in this enzyme mutation diabetes mellitus. Diabetes mellitus. Diabetes mellitus. Diabetes mellitus. Diabetes mellitus. Diabetes mellitus. Okay. صار in general في الميديسن يقولوا class 1 class 2. لكن class 2 yes sensitivity to insulin decreased. لكن في حقيقة الأمر احنا على مستوى المولكيول لا يدرس في الطب أو في الهوسبيتال وين البروتين defected وين الانزيم defected وين okay. And the sensitivity of insulin may be defective receptors or signal transduction. Yes? Or maybe due to adiposis. Adiposis decreasing the sensitivity to insulin. Okay, صح؟ لكن لو ما فيش insulin or insulin deficiency diabetes mellitus class 1. Okay. شوفوا في الجوجل DPP4 inhibitors drugs تعرفوا عليها من فارماكولوجي. DPP4 dipeptidase 4 and GLB1 yes agonists. جوجل تلقى ليستة من الدراجز. هتلقى في بعض منها موجودة في الفارماسي. موجودة. هذه last drugs for treatment of diabetes mellitus. يعني ممكن في 2005 بدت ريليزت. يعتبر very new. Okay. نجو إلى الجلايكوليسيس.